The last speaker is Dr. Evgeniya Schastnaya from East Coast Medicine, Baltimore. The title is using signaling pathway activation analysis to identify prospective drugs that may be used for treatment of androgenetic alopecia.
Thank you very much. I hope you have some energy left. My name is Evgeniya Schastnaya, but please feel free to call me Jane. I'm a junior scientist at a bioinformatics company called In Silico Medicine. It's located in Baltimore at Johns Hopkins University. I have nothing to disclose. In silico medicine is utilizing advances in genomics and big data analysis for in silico drug discovery for age-related diseases and cancer. We developed and utilized several packages for aging and cancer research and provide services for pharmaceutical and cosmetics companies. Primarily and originally our company was focused on cancer research, and after we validated our algorithms for cancer, we expanded to age-related diseases and aging in general. Recently we've launched several projects on skin aging and photo aging, and now we are here to understand how we can contribute to hair research. So it is obvious that thick hair is associated with young age, and thanks to your hard work, our understanding of complex molecular signals and mechanisms that influence and regulate hair follicle cycles expanded in recent years, but there is still much work to do. As it has been previously said many times during this wonderful congress, we need to understand the undergoing processes on the pathway level to develop new therapeutic targets and approaches. Additionally, as a group that studies aging, we believe that hair follicle may be an easily accessible tool for gerontology. So in silico medicine is interested in identifying pathways affected in different types of alopecia to develop rational targets for therapy. In this experiment, we used publicly available dataset from Gene Expression Omnibus. This dataset contains gene expression data for cells from biopsies obtained from both haired and bald parts of the scalp of 5 individuals with androgenetic alopecia. We used our own platform called OncoFinder to identify pathways that are dysregulated, and then we identified candidate drugs that can potentially cure the disease state. So how does this work? OncoFinder tool has the ability to elucidate and precisely define altered features of intracellular signaling using mathematical computations. As an output, this algorithm produced pathway activation strength values, where positive and negative PAS values correspond respectively to up and down regulated pathways. The PAS is a measure of the cumulative value of perturbations of a signaling pathway and may serve as a distinct indicator of pathological changes in the intracellular signaling machine of the cell or even tissue level. This approach has been previously suggested to study aging and to rank candidate geroprotectors, and also signaling pathways were shown to be better markers for cancer than expression of individual genes. The next step in developing new therapeutic strategies is drug scoring. We developed a computational method for discovering new uses of existing drugs based in pathway activation analysis. We have a large database of drugs, and we know their effect on gene expression on cells, and by analyzing dysregulation of signaling pathways in the disease sample compared to the norm, we perform drug repositioning. We calculate special values called drug scores for each drug, so that the higher drug scores correspond to drugs that can better mimic pathway activation level and potentially cure the disease state. So here are our results on pathway activation analysis and drug scoring for this particular sample. These are just several pathways that are dysregulated. They may be the most famous ones, like TGF-beta, as I know, is quite controversial because It mediates the reduction of catagen while it is essential for the start of anagen phase. And as for the drug repurposing, as I know, these top drugs have never been used for treatment of androgenetic alopecia, but we also showed that this famous JAK-STAT inhibitor has this drug score according to our system. So in silico medicine is interested in studying the differences in signal and pathway activation levels in androgenetic alopecia and senescent alopecia to develop individual treatments of hair loss for people with different ages. We are also interested in studying other kinds of alopecias, as well as characterizing senescent alopecia signaling pathways to understand systemic senescent processes. So if you perform gene expression analysis, don't hesitate to contact me because our academic collaboration may result in identifying new targets for therapies, and together we can make a huge contribution to this field. Thank you very much.
Yes, so we finished 4 papers and we have still several minutes left for discussion. Is there any question? Yes. Number 1.
I have a question for Dr. Biliardi. I was struck by a small number of genes that you actually saw changed in the AGA patients, and if I understood correctly, you performed the RNA-seq with total preparations of the follicle. So I would worry that you have low detection sensitivity for genes in the DP, for instance, if they play an essential role and are changed because the DP is only a small fraction in the preparation.
So it was not the full hair follicle, it was only the hair valve. We got very high quality and enough RNA, but as I told you, our bioinformatician put on a very rigorous program. So we got several hundreds, I think 2,000 or 3,000 genes changed, but these were the most significant ones.
Dr. Biliardi, your data came from biopsy hair follicle?
Follicle unit extractions, yes.
Biopsy, not plucked hair?
No, not plucked hair. Follicle unit extractions.
Any other question? Dr. Ramo, may I ask one question? Your stable amine is also active and effective in animal model?
In that you ask if the polyamines are also effective in animal models? Yes, there are many in vivo models using animals. Actually, everything started with animals, and we try to compare that to humans. But in animals, there are a lot of data to show that polyamines can also go here.
I also have a question for Dr. Rama. Do you have an hypothesis about how these different mechanisms can interact between each other to prolong anagen, or if there is a leading mechanism in your model?
Well, as I showed I think it was one of my first slides, that polyamines have a plethora of effects in cells, not only in human hair follicles, but they're important for many different functions, and I believe they work in parallel. I don't think there is one pathway, but polyamines are interacting with many different mechanisms in the cells, and I don't think that it's one single mechanism can connect them all.
Well, thank you very much. I'm surprised this many are still here at the end of the last session. Dr. Stroll and I would like to thank the presenters. We'd like to thank all the people who organized this meeting. Hope you have a safe trip home and enjoy the meeting. And now?
Please do not go. We are going to have the closing ceremony.